First of all, I want to thank Gabriela and Maribel for organizing this very interesting meeting and in this beautiful place. So it's a pleasure to be here with all of you and to share what we have been doing. Okay? So, Darwin had an abominable mystery. No? He wrote that the rapid development, as far as we can judge, of all the higher plants within recent geological times is an abominable mystery. So that was Darwin's. No? So now I want to show you my own abominable mystery. And it's this one. So you are looking at a small DNA sequence made of 8 nucleotides that is called the Highly Iterated Palindrome 1. That, as you can see, is palindromic, as the name says. But what is so peculiar about this sequence? Okay, what is so interesting, so different? The thing is that this sequence is really highly abundant in cyanobacterial genomes. So what you are looking here is at the abundance, the frequency in this axis, and here the overrepresentation of all octameric palindromic sequence in cyanobacterial genomes. So as you can see, you have a big cloud here of several octameric palindromic sequences, and then you can see this small cloud in color red. You are going to see the HIP1, the highly iterated palindrome 1, and then with some other colors, you will see other palindromic sequences that are also highly abundant. So why these sequences are so abundant? What are they doing? This is only a fragment of one of the genomes from a cyanobacteria called Synechococcus elongatus, where you can see you have here about 3,000 base pairs, and you can see how common these sequences are along the genome. Huh? So what are they doing there? Okay. Well, what is surprising is that despite their abundance, the function of these sequences remains poorly understood, including their origin. So this is, I think, a very interesting mystery to try to solve. Well, okay. What else do we know about these sequences? Well, when they were first characterized in the 1990s, it was suggested that highly iterated palindromes like HIT1 are an adaptation for genome plasticity by promoting intramolecular recombination repair. In a similar way as the Inococcus radiodurans repairs its genome, it is known that it has several genome copies and use those copies to repair the genome, and that way it can resist the desiccation and radioactivity. Several cyanobacteria also have several copies of their genomes. So maybe these authors speculated they were using this small sequence to repair the genome by homologous recombination. In fact, very early in the 1990s, in 1999, there was some evidence that this sequence was a resolution site for site-specific plasmid recombination. So there is some evidence that it can be used for recombination. Also, interestingly, this sequence is abundant in most, but not all cyanobacteria. Here you are looking at a phylogenetic tree of cyanobacteria. Here you have the oldest cyanobacteria, which are called Bleobacter. And then you can see that with the green bars, several species of cyanobacteria have this sequence in high frequency. And in orange bars, you can see that they are also enriched in this sequence. But you can see also that not all cyanobacteria show this phenomenon. So for instance, marine picocyanobacteria, which are those ones, are not enriched in this motif, and those are maybe the most common cyanobacteria in the world, which are in the oceans. And there are also some other species of cyanobacteria that do not show this phenomena, for instance this one, and some others along the tree. Also, very interesting, and I think this is very important. HIP1 sites are conserved among cyanobacteria. If you align two genomes, for instance, the genome from these two species, and you look for the homologous sites along the genomes, you will see that in different pairs of species, the percentage of HIP sites that are conserved along the genomes are more conserved than control sequences. No? So you see this pattern in the coding region as well as in the non-coding region. So I think this is very, very important because conservation is the hallmark of function. So if it is conserved, then this sequence must be doing something. No? For something, it's, the cell has this motif conserved. Well, also, very interestingly, this sequence is methylated in two sites. Here you have HIP1, and one DNA methyltransferase methylates the A by recognizing these four bases, and another different DNA methylates the cytosine by recognizing these sequences. So those sequences are the target of DNA methylases. And for instance, in this cyanobacteria, Cynecocystis 6A03, which is a model cyanobacteria, 85% of all the sites are methylated in the system. So they are methylated. Not all the sites. And this is important because it is also known that this methylase is not part of a restriction modification system. Because there are no activities of restriction endonucleases coded in this cyanobacteria that targets specifically this site. So it is not part of an RM system. Okay? So this is important also. Well, also very interestingly, and I think this has to do this points to the function of this motif, is that if you want to transform, for instance, a cyanobacteria, in this case again, Cynecocystis 6A03, if you add to the plasmid this sequence, and if you methylate that sequence, the efficiency of transformation increases. Okay, so this is very interesting, and it is also known that in another different species of cyanobacteria, Synechococcus 7002, the same phenomena happens. So if you methylate this sequence, the efficiency of transformation increases, and if you include the sequence without methylation, also the efficiency of transformation increases, differing if you do not include this sequence. So I think this is also very interesting and tells something about the function of this sequence. So let me recapitulate so far. HIP1 is abundant in most, but not all cyanobacteria. HIP1 is evolutionarily conserved, so it's likely to have a function. HIP1 tends to be methylated. It is not part of an RM system. By itself, it increases the rate of transformation, and in particular, when methylated, it increases the rate of transformation even more. Okay? Well, so very recently, these authors proposed, I don't have much time to go into the details that led these authors to propose this, but they suggested that this sequence improves transformation efficiency via two complementary mechanisms. First, by altering DNA binding uptake processing prior to homologous recombination, and second, by increasing the efficiency of homologous recombination in a manner reminiscent of a crossover Chi site. Okay? So these two mechanisms may help or make use this sequence to increase the transformation rate. So we have an hypothesis, and the hypothesis is that HIP1 may function as a DNA uptake sequence for cyanobacteria. That That is, this helps cyanobacteria to identify self from non-self DNA, or perhaps friendly from unfriendly DNA in the medium. If this is true, then there must be some proteins that recognize and bind specifically to this sequence. So, the question related to the hypothesis is whether there are protein families whose phylogenetic distribution coincides with that of HIT1. The rationale is that those protein families that have a phylogenetic distribution coinciding with that of HIT1 abundance will be functionally related to this highly abundant palindrome. Okay? So this is the idea. No? In the tree of life, if you find, for instance, some cyanobacteria which have this phenomena of a highly abundant motif, and you find some protein families that relate to this abundance, and you do not find those proteins in those cyanobacteria that do not show high HIT1 abundance, then maybe there is a functional correlation. So the methodology, straightforward, is that we went download the genomes from about 100 cyanobacteria. We identified the protein families between those genomes, and then we asked whether there is a correlation between the abundance of the palindrome and the phylogenetic distribution of the protein families. And of course, to measure the correlation, we use a method that is phylogenetic aware in order to take into account the phylogenetic structure of the data. Okay? Well, these are the results. The results are from my previous student, Ulises Rodriguez, that now is part of Delaye and Luis' group. And here you have the phylogenetic tree of cyanobacteria. We use here you have the names of the species of cyanobacteria. Then in the first 6 columns, of course you may not see this, but well, we classify cyanobacteria as having or not having a highly iterated palindrome. And then here, each column is a protein family. So this table goes far away to the next block, because you have all the protein families that are conserved in these genomes. But I'm showing just the first part of the table. And okay, what we found? We found 3 protein families that match very closely to the phylogenetic distribution of the highly iterated palindromes. Okay? So these proteins are the tetratricopeptide repeat protein, the G-domain containing protein, and a chemetaxis protein GW. Okay? So the question is, do they bind to DNA? So we were able to do this a few years ago when Ulises was doing the master in my lab, in my group. But now we have AlphaFold 3, so we started to play with this tool. And this is really just playing. This is very recent data. It's just, we were learning to use the tool and just playing with it. And what we found, interestingly, for the tetratricopeptide repeat protein, is that there is, according to AlphaFold 3, some evidence that it may bind HIT1. This is shown here in this chart by these green boxes in these places, which suggests an interaction between the protein and the DNA. And interestingly, the 2 amino acids, histidine and proline, which are close to the mallyl group, where you have the modified methylated bases, these amino acids are also conserved in the multiple alignment. So this correlation is very, very interesting. Of course, the quantification of the model is not very good. Those values must be over 0.5, so the model is not that good. But anyway, for this first data, we were very happy. No? So this made some sense of this. This is the data for the G-domain containing protein, also in AlphaFold 3. There is again some evidence that there may be an interaction. the model is also not very good. If you see this color yellow here, it's that for the model is not very good for the DNA. You see a pair of lysines pointing towards the major group, and these 3 amino acids, the tyrosine, arginine, and leucine, are also conserved in the multiple alignment, although they are not pointing directly toward DNA. So it is interesting also. And finally, for the chemotaxis protein GW, there is no evidence of interaction between alpha-4-3 and between the proteins and the DNA. Okay? But anyway, what is interesting about this protein, GW, is that there is genetic evidence also in Synecocystis 6803 that this protein, altogether with other proteins, are more or less required for pilus assembly, motility, and natural transformation with extraneous DNA. So there is some experimental evidence of the interaction. Okay, as conclusions. There are 3 proteins whose phylogenetic distribution coincides with that of HIP1 and other iterated palindromes. Those are the tetratricopeptide repeat protein, J, and the J domain-containing protein, which may link to HIP1. The chemotaxis protein GW may participate in pilus assembly and natural transformation competence, but further in vitro experiments are required. And I'm trying to combine some experimental colleagues in order to do the experiments in the lab. No? So to see if these observations hold. I think it could be interesting, the working hypothesis, that this sequence may be used by cyanobacteria to identify self and non-self DNA. So maybe it's an adaptation for these microorganisms. And I want to give special thanks with my previous students, Ulises and Eduardo, who have been working on this project, and to collaborators to whom I have been talking about this project, and I hope to get new friends here to continue collaborating in this subject. So thank you very much, and I'm ready to take some questions.
I have dos preguntas and 2 questions. 3 questions, very quick questions. So my quick question and comment. One is that we found, because I'm also fascinated by repellents, in the nitrogen fixation cluster that I was talking about, it's 23 kV, there's not a single repellent. So that's just maybe a clue. And then the other thought I had is from a long time ago that these hip elements seem to be at particular distances, they're not too close to each other. The fact that you link them to the tripeptide domains, I'm wondering if, because these chromosomes have to be wound around, especially if they're multiples, could it be a site for looping?
Yeah. Yes, one of the, I didn't show that, but one of the things that are known is that these sequences seems to be repeated, not randomly, but periodically.
Yeah.
So it has been suggested that they may help the chromosome to fold somehow, no? Yeah.
Thank you for the great talk. In other systems, other microbial systems, bacterial systems, when you see these specific sequences that are associated with horizontal gene transfer, they tend to be very species specific, right? And yours is like about as far away as that is almost possible. So that's a very interesting contrast, and I was wondering if you could comment more on that.
Okay, it's a good question. So far, we have found this sequence only in cyanobacteria, not in other palindrome. No? So I don't know why it's so particular for cyanobacteria, like a cyanobacterial mark.
It's a really interesting puzzle that they're trying to solve. With so many gene families and so few comparatively genomes, you would expect lots of correlations just by chance, right? Did you study the expected distribution or a known model of the correlations just by chance?
Well, the model for the abundance of the palindromes calculates an expected frequency of observations. But maybe that's not the thing you're asking.
No, I mean just about the correlations. When you have when you correlate a data set with so many other variables, you're going to have some by chance. Because how many gene families were there?
Yes.
Thousands?
Yes, thousands.
And only 100 genomes.
Yes.
So you would absolutely expect lots of correlations just by chance, right?
Yes. In fact, we eagerly look for cyanobacteria genomes that do not have this motif and are in another part of the tree. So you can increase like the resolution of the analysis. No? So far we have very few clades without HIP1, and because of that, the chances that the correlation is, spurious, is higher. So if there are some genomes that do not have the HIP1 and are not the known, they do not belong to the known clades that do not have, it will be very important for us. No? So maybe here in Cuatro Ciénegas or somewhere else, no? It will be very helpful for increasing the resolution. Yes. Yes.